In this video, we're going to talk about fatty acid synthesis. And by the time you're done watching it, I want you to be able to answer these questions. What part of the cell is the source of acetyl-CoA? Where does fatty acid synthesis occur? What is the net effect of moving an acetyl-CoA to the cytoplasm from the matrix? How are acetyl-CoAs activated for the production of fatty acids? What is special about fatty acid synthase? What reactants are necessary for one round of fatty acid synthesis? And what is the basic chemical strategy involved in one round of fatty acid synthesis? All right, the basic unit of production of fatty acids is acetyl-CoA here. And that is in the mitochondrial matrix, but fatty acid synthesis occurs within the cytoplasm. And so we need to get acetyl-CoA from here in the mitochondria to here in the cytoplasm. The problem is it cannot get through that inner mitochondrial membrane on its own. But there is a citrate transporter. So what happens is there's first a reaction in which acetyl-CoA binds to oxaloacetate, and that becomes citrate. So now this citrate can move through the citrate transporter out into the cytoplasm and then undergo the reverse reaction in which it releases an acetyl-CoA and oxaloacetate. And the problem is, okay, although we have acetyl-CoA out here, we didn't want to move an oxaloacetate out there, so we need to get oxaloacetate back into the mitochondria. But there is no oxaloacetate transporter within the mitochondrial membranes. So what happens instead is this oxaloacetate is first reduced to malate with the electrons from NADH, and then that malate is oxidized to pyruvate. But the electrons and hydrogens from the malate oxidation here, don't go back onto an NAD+. Instead, they go to an NADPH+. And remember, NADPH is always involved in biosynthesis, not oxidation. So this is going to be the source of hydrogens and electrons that we're going to use to reduce our fatty acids. Now the byproduct of this is pyruvate, which will slip through the pyruvate transporter and again be turned into oxaloacetate. So the net effect of this transporter system is to move one acetyl-CoA from the mitochondria to the cytoplasm and then convert one NADH within the cytoplasm to one NADPH. And the nice thing is both of these, NADPH and the acetyl-CoA, are going to be necessary for the fatty acid synthesis. Now, acetyl-CoAs do not have a high enough energy to be able to be put together into a fatty acid. You have to activate acetyl-CoA first. And the way that occurs is through an enzyme, a reaction catalyzed by this enzyme here, acetyl-CoA carboxylase, ACC. What that does is it takes this bicarbonate here and it uses it to add a carboxyl group to the methyl group of acetyl-CoA. So what we have here is what's called Malonyl-CoA. It's our acetyl-CoA that's had a carboxyl group added to it. And now this is what's going to be used to make fatty acids over and over and over again in 2 carbon units. Why 2 carbon units? Because this carboxyl group was there just to activate the molecule, and the very first reaction of fatty acid synthesis is the decarboxylation of that malonyl group and the addition of those 2 carbons on to a pre-existing acetyl-CoA. So let me explain that a little bit better here. What we have here is fatty acid synthase. And fatty acid synthase is pre-primed with an acetyl-CoA right there. Okay, so this is before the production, before any rounds of fatty acid synthesis has occurred. So this is going to be the first round. And incoming here is our first Malonyl-CoA, our malonyl group that's going to be added. What we see is it's going to be This carbon is going to attack this carbon here, this carbonyl carbon. That's our thioester bond, right there. And we are going to lose from that the carboxyl group that we just added. Right? So it was simply there just to activate acetyl-CoA. And now what we have is that two carbons from acetyl-CoA that had pre-primed this are now attached to the two carbons of what was once malonyl-CoA. And so here we have our growing fatty acid chain. This is what is normally the carboxyl end, which means the next carbon is the alpha, and then the third carbon down is the beta. And so now instead of oxidizing this beta carbon, we are reducing it, which means, let's take a look at it. This is the reversal of what had happened in beta oxidation. We have a carbonyl, so we're going to first then reduce it. Where's the source of electrons and source of hydrogens? NADPH, right here. So now we went from a carbonyl to a hydroxyl. Next, we're going to then convert that hydroxyl group into an alkene, okay, by taking out a molecule of water. A dehydration reaction. Remember, this is the opposite of that hydrolysis reaction where we added the water into it, or the hydration reaction, not the hydrolysis, but the hydration reaction. Okay, so we pulled out a molecule of water and we went from an alcohol to an alkene. All right, next here's that alkene, we want to reduce it further. How do we reduce an alkene? We turn it into an alkane. Go from a double bond to a single bond. Where do those extra hydrogens and electrons come from? NADPH, right here. And so we go take those electrons from the NADPH and the hydrogens, and we convert that double bond to a single bond. And now we have completely reduced this beta carbon here. And we are ready for a new round, a new malonyl-CoA to come in and then be added onto this chain. So we start, absolute start, a one-time deal. We start with an acetyl-CoA that's primed on fatty acid synthase. And then each round of fatty acid synthesis is going to involve one molecule of malonyl-CoA and two molecules of NADPH. The products then are, per round, one CO2, that's the byproduct of the malonyl-CoA, two expended NADP pluses, and our fatty acid has increased in size by 2 carbons. Now the molecule, the enzyme that catalyzes all of these 1, 2, 3, 4 reactions actually exists in a single polypeptide chain. A single polypeptide chain that has 7 different domains, each domain acting with specific catalytic activity. So it's pretty incredible. It's a complex enzyme, a complex protein. Now, the maximum size fatty acid that we can get from this enzyme is 16 carbons. Any fatty acids bigger need another set of enzymes to elongate it further or to unsaturate it to add double bonds. But those are enzymes that are not necessary to know for this course. All right, so again, make sure you go back through that video, you review it several times, and make sure that you can answer each one of these questions.